So welcome to part 2 of the digestive system physiology. This is when we start talking about the physiological mechanisms associated with digestion of nutrients as well as absorption of that nutrients. So digestion and absorption, those terms have different meanings and oftentimes students get them mixed up. The term digestion means to break down the food. You're going to break it down into smaller and smaller units until you reach the monomeric unit, in which you will then absorb the food. And absorption just means you're going to take that nutrients that you digested and you're going to move it across the wall of the small intestine and into the blood supply. So most digestion and almost all absorption is going to happen after the chyme goes into the small intestine. So it's in the small intestine that these two processes predominantly happen. Now, a lot of students think that the stomach is the place where digestion and absorption happens, but that's incorrect. Remember, the main job of the stomach is to act as a storage facility for that bolus that's entering in and turning into chyme. Of course, the stomach does have other functions as well, but that was one of the predominant functions of the stomach. There are certain things that can be absorbed in the stomach, and those things include aspirin, some lipid-soluble drugs, as well as some alcohol. So let's start at the stomach, and then we're going to move our way to the intestines, the small intestine. How does the stomach protect itself from the acidic solution inside? Well, remember, the pH of the stomach is a pH of about 1 to 2. That low of a pH can damage human tissues very easily. So what makes the inner lining of the stomach so special? Well, there are 3 main things that actually make that lining special, and it's going to include having a mucus coat on the inside of the stomach. This mucus coat is not like any mucus coat. It is a very alkaline mucus coat, which means it has a very basic pH, so that when it does interact with the acidic juices inside the stomach, it neutralizes them. The second thing is the tight junctions between the epithelial cells inside of the stomach. This prevents the acidic gastric juices from leaking out to the rest of the cavity of the body. And the third one is the mere fact that it does get damaged. The inner lining of your stomach does get damaged from the acid. But no big deal, because those cells inside the stomach actually live only 3 to 6 days before they're recreated and they slosh off and they go and mix with the chyme. So there is recreation of damaged, recreation of cells when those cells get damaged. However, if any of these 3 measures end up not working, they break, you're not able to make a mucus coat, the tight junctions aren't so tight anymore, that can cause inflammation, ulcers, tissue damage, and if left untreated, it can cause a GI bleed. It can cause bleeding into the digestive system from the vascularization around that digestive system. So how do we regulate how much acid we make? When the stomach churns, how quickly the stomach churns. Well, this is all under regulation of gastric function. In the next few slides, your job as a student is to know the names of the peptides that I'm going to give you and their functions. These peptides are part of a group called gut-brain peptides. They're just peptides or little proteins that interact between your gut and your brain. There are short-term regulators of appetite and long-term regulators of appetite. So there are short-term regulators of appetite, meaning there are peptides that control your immediate hunger and satiety. And then there are other peptides that can control long-term hunger and satiety. So peptides that actually contribute to obesity. The first short-term peptide is called ghrelin. This is a peptide that we actually talked about in the first digestive system PowerPoint. And ghrelin is released from the stomach when the stomach is empty. So when the muscle around the stomach, is not stretched out, it is empty, there's no distension in there, stomach is going to make ghrelin. Ghrelin will go to your brain to tell your brain, hey, the stomach is empty. You're hungry. And it'll signal the brain to produce other hormones, and it's going to start at the hypothalamus. So ghrelin signals the hypothalamus to make something called growth hormone releasing hormone. I'm going to underline this because this is an important hormone name to know in terms of its association with ghrelin. So when the stomach makes ghrelin, the ghrelin signals to the hypothalamus makes GHRH, and growth hormone releasing hormone eventually causes the production of growth hormone, and growth hormone can make you feel hungry. This can also increase production of something called NPY. We will talk about this a little bit later on. For now, keep it in your back pocket. Ultimately, the major goal here is to increase appetite to fill this guy up. The next short-term regulator of appetite is called peptide YY, abbreviated as PYY. This peptide is secreted by enteroendocrine cells of the ileum and the colon. The ileum is part of the small intestine, the colon is part of the large intestine. So both intestines participate in producing peptide YY through their enteroendocrine cells. Just as a side note, these are cells that are found in the lumen of those regions. So, your body can sense when food has arrived in the stomach. Usually that's because the stomach gets stretched out, the pH of the stomach rises. So when this chyme in the stomach fills the stomach and stretches it out, you're able to secrete PYY. And you're secreting PYY to tell your brain, hey, we received the food that you ate and you can stop eating. It's there to signal satiety. So what you see on these pictures, and I think this will clarify things a bit, is when food is missing from the stomach is not stretched out and it produces ghrelin. Ghrelin signals your brain to be hungry. When you do have food in the stomach, eventually this chyme makes its way into the small intestine and into the large intestine. When that chyme gets to the end of the small intestine, the ileum, and the beginning of the large intestine, the colon, these two regions of enteroendocrine cells are going to produce PYY. You do see two other peptides here. We're going to talk about CCK later, but just for now, let's keep it to PYY. So once you secrete the PYY, the job of PYY is to, again, go to your brain, but this time, if you see the red X's, this is a down regulator. PYY tells your brain that you are full now, so it signals satiety. So we have ghrelin signaling hunger, PYY signaling satiety so far. Both of these are short-term regulators, so they work immediately, and then they can stop their function pretty quickly as well. The third short-term regulator is CCK. If I go back a slide, that's this one right over here. CCK is again secreted by enteroendocrine cells, the same inner lining of cells, but this time from two other parts of the small intestine, the duodenum and the jejunum. CCK is produced when chyme enters those regions. So this greenness chyme, it's going from the stomach into the duodenum. When this duodenum senses the incoming chyme, it makes CCK, and I'm going to get my pen out here. It makes CCK to tell the gallbladder to secrete its bile, and the job of bile is to break down fat. And it also sends that CCK to the pancreas to tell the pancreas to secrete proteases. These are just enzymes that are part of digestion. So the pancreas makes a slurry of enzymes that have to do with digestion of nutrients, and we're going to go really in depth in this a little bit later on in this PowerPoint. But for now, when chyme enters the duodenum, the enteroendocrine cells in the duodenum secrete CCK, And that CCK signals to two places. Number one, the gallbladder to secrete bile, and number two, the pancreas to secrete digestive enzymes. The third place that's not shown on here, and we'll draw it over here, and hopefully this is going to be a decent enough drawing, is your brain. Let's draw the spinal cord out there. So we got the ventricle there too. CCK is going to send a signal to your brain to say, hey, we're full. So red X. So it's going to suppress your appetite. It signals satiety. Why? Well, because the food's already in the duodenum. You've eaten. We're now on the process of digesting that food. So three places that CCK signals: gallbladder, pancreas, and the brain. Now moving on to long-term gut-brain peptides. These peptides can affect your hunger and satiety from weeks to years. You've got leptin and insulin. These are the only two that we're going to talk about in this PowerPoint, but I do need you to know the names and the functions again of these two. Leptin is secreted by adipocytes. You have the prefix adipo in here. That means fat. So the fat cells that you have will make leptin. Leptin is produced based on the proportion of how much fat you have. So the more fat you have, the more leptin you produce. The whole point of this is to inform your brain of how much fat reserve you have. So the more fat cells you have, the more leptin you make. And that signal basically is to tell your brain, hey, we have enough fat reserves. Stop storing nutrients as fat. And it also triggers satiety. Leptin is where there's a lot of research for obesity. So obesity is a disease in which you're not getting the signals of satiety. So people who are obese are more likely to have defective leptin receptors in their brain. So you can make a ton of leptin, but if you don't have the receptors in your brain will never receive the signal that you have enough fat storage. So people who are obese are constantly feeling hungry, and they're constantly storing their nutrients as fat because their brain doesn't think they have enough fat storage. Insulin is another long-term gut-brain peptide. I know a lot of students think, oh, insulin, it's just to regulate sugar levels. Well, yeah, you're right. Insulin is secreted by pancreatic beta cells to stimulate glucose uptake and lower blood glucose levels. But this also can send a signal to your brain to tell your brain that you are full. It's not as strong of a signal as leptin is, so it's weaker appetite than leptin, but nonetheless, this does send a signal to your brain to tell your brain that you're full. Now, we're going to go one step more in depth. If at this point you'd like to pause the video and go back and review those short-term and long-term gut-brain peptides, this would be a great place to pause the video and get familiar with those 5 peptides, because this is where we're going to tie it all in together, and it can get a bit confusing. So we'll go through the written portion of it, and then we have a wonderful illustration to look at. So, regulating your appetite at the hypothalamus. In the hypothalamus, there are 2 neural networks in that region. One is going to be a region that stimulates appetite. The other region is going to be a place that suppresses appetite. One of the regions will secrete something called neuropeptide Y. This is secreted by the hypothalamus to stimulate hunger. When you make this, you feel hungry, so it is an appetite stimulant. Ghrelin was one of the short-term gut-brain peptides that we talked about that actually promotes hunger. So ghrelin stimulates NPY. So I'm going to draw an arrow from ghrelin to NPY, and I'm going to put a plus sign next to it. Insulin, PYY, and leptin, all 3 of these, as you now know, are going to stimulate satiety. All 3 of these are going to downregulate NPY. So the more of ghrelin you make, the more you stimulate NPY, And this NPY is what goes to the cortex of your brain to consciously tell you you're hungry. The more insulin, PYY, and leptin you make, the more you downregulate production of NPY so that you don't tell the cortex of your brain that you're hungry. Now, the other region in the hypothalamus is going to secrete something called melanocortin. Melanocortin inhibits eating. This says, hey, you're full. So if you're signaling melanocortin, you're going to signal that you're full. Now we're going to see all of this in this illustration. I would highly suggest you star this slide. This is a great slide to star. What we see here are the different regions of the digestive system, what peptides they secrete, and the role of those peptides in the hypothalamus. This is just a very zoomed in region of the hypothalamus. Of course, it's not this big in the real brain, as you know from anatomy. So let's go one by one. This slide could be used as a review for a lot of you who've already gone through the previous slides. Your pancreas will produce insulin in the presence of sugar. Insulin's job is to downregulate NPY. If you downregulate NPY, you are not going to signal hunger. In fact, you are signaling satiety as a byproduct. Then you have the stomach. When the stomach is empty, it makes ghrelin. Ghrelin is a hunger peptide, so naturally it's going to upregulate NPY to then signal hunger in the cortex of the brain. Now, before I move on any further, I'd like to clarify one thing here. I'm going to clarify this pathway right over here. If you upregulate NPY, this automatically sends a downregulation signal to melanocortin. So if you upregulate hunger, at the same time, you're downregulating satiety. Downregulating NPY just does that. It just downregulates NPY, nothing else. And then you have another signal that can upregulate melanocortin. So let's move on to the next one. The small intestine, when it senses that there's food, will make PYY. PYY signals satiety. It downregulates NPY. And if you downregulate NPY, you're not going to feel hungry. And as a byproduct, melanocortin signals satiety. Small intestine also makes CCK. CCK downregulates NPY, which means we're not going to signal hunger. And because we're not signaling hunger, we're not going to downregulate melanocortin, and this guy is just constitutively going to be on. Then you have the fat cells, the adipocytes. The more of these you have, the more leptin you make. Now, leptin is kind of fancy because leptin does both at one time. Leptin can downregulate NPY to inhibit hunger signals, but it can also at the same time upregulate melanocortin to signal satiety. If you notice, this is one of the only peptides that is directly going and upregulating melanocortin to signal satiety. So imagine, if you can't receive the signal from leptin in your brain, you cannot upregulate melanocortin, and you will not be able to feel satiated. Imagine never feeling full. Even though you are making the peptide to signal the brain to feel full, you're missing the receptor for it up here. So if you are confused on this slide, please message me for office hours. I would love to go over it with you guys and explain it to you one-on-one if it helps. So we're moving on to gastric phases. So we did regulation of hunger, and that had to do with stomach and production of gut-brain peptides. Now we're going to the different gastric phases. There are 3 of them. Gastric phases are the phases that you go through to get the stomach activated, to get it functioning, and then to deactivate it after the food has already moved onto the small intestine. The first phase is called the cephalic phase. Cephal means head. The cephalic phase is actually the phase where you're just thinking about food, smelling food, you're looking at food, and all of that sends a signal to your brain to say, hey, we're about to eat. And the brain says, okay, let me get the stomach ready. So the hypothalamus in the brain sends a signal down to the medulla. Medulla will trigger the vagus nerve. Remember, this is your parasympathetic nervous system fiber. It's the rest and digest system. The word digest is in there. The vagus nerve, so far you've learned that it innervates the heart. What have you learned that the vagus nerve does to the rate of the heart? It lowers the heart rate. What you haven't learned yet, though, is that the vagus nerve actually innervates a lot of other places in your body, too. One of those places is going to be the stomach. The vagus nerve signals the stomach to increase the gastric secretions. This includes stomach acid, and it includes pepsinogen. We talked about these in the first PowerPoint. The second phase is called the gastric phase. This is when the food has been eaten, it's been swallowed, and it drops down into the stomach. Now, the food is going to, number one, stretch out the stomach. And number two, it's going to increase the pH of the stomach because normally the stomach's pH is about 1 or 2. And I'm hoping the food that you eat is not at that pH. That's a really, really acidic meal that you're eating. The pH of the food that you eat is normally around, I would say, between 5.5 and maybe 10. So the overall pH of the stomach increases. These are two changes that are sensed by the stomach, and those changes will cause the stomach to produce even more gastric secretions. In addition to these two signaling production of gastric secretions, there are 3 other chemicals that actually signal the production of gastric juices. Number 1, acetylcholine. Remember, this is the parasympathetic neurotransmitter. When you are in your parasympathetic response, the rest and digest, the vagus nerve secretes acetylcholine onto the stomach to get the stomach to start functioning. Histamine is also produced by enteroendocrine cells in the gastric glands. That's going to send a paracrine signal. So it's a signal from a cell to a very close neighboring cell. The cells that we're talking about are in the gastric gland, and they're going to signal cells that are in the stomach as well to start making more gastric secretions. The last one is called gastrin. If you remember from the last PowerPoint on digestive system, we talked about gastric glands, and we talked about chief cells, we talked about mucus cells, parietal cells, and there was another cell called a G cell. We didn't talk about that yet, but this is where we're going to introduce it. G cells in the stomach make a hormone called gastrin. Gastrin self-regulates the stomach by signaling back onto the stomach itself and telling it to upregulate itself. So in addition to these 2 signals triggering production of gastric juices, you do have 3 chemicals that are also going to take part in upregulating the stomach and gastric secretions. The third phase is called the intestinal phase. Like you might imagine, this has to do with the intestines. When food goes from the stomach into the intestines, the acidity of that chyme as it goes into the intestines is a trigger. Acidic chyme is going to lower the pH of a normally basic intestine. Your small intestine, the duodenum specifically, likes to be around a pH of 9 or 10. So when acidic chyme, usually at a pH of like 3 or 4 now, enters into that duodenum, that sends a chemical signal to say, hey, we've received the chyme from the stomach. So because of presence of acidic chyme, as well as stretching of the duodenum, we're going to begin the intestinal phase. So this one's a little bit weird because the duodenum initially at the first presence of this acidic chyme, upregulates the stomach to say, okay, I now know that the stomach is sending over some chyme to me. Let's get that stomach to continue working so it can churn and keep pushing the rest of the food into the intestines. So we initially upregulate the stomach, but then after maybe a few minutes, the small intestine will downregulate the stomach. There are two main signals that cause the duodenum to initially upregulate the stomach. Number one, the stretching of that duodenum. Number two, and this is a new one, partially digested proteins from the stomach going into the small intestine. And these partially digested proteins, if you recall, are partially digested because the stomach makes pepsinogen. Pepsinogen gets activated into pepsin, and the job of pepsin is to partially digest long chains of proteins. So once you partially digest long chains of proteins, those smaller peptides enter into the duodenum, and those peptides themselves signal the G cells to make even more gastrin. If I go back a slide, we saw this in the gastric phase as well. G cells make gastrin to upregulate the stomach. This is in the presence of food in the stomach. So one signal for G cells is the stomach stretching. Another signal for G cells is going to be presence of peptides. Now here is a note that you have to write down. There are G cells in the stomach, and there are G cells in the duodenum. They're G cells that both produce gastrin, but they are found in different places. So kind of wrapping this up in terms of gastric function, we are able to regulate gastric function in four main ways. So we're going to tie in a few different things together right now. Number one, you have what's called the enterogastric reflex. Because you bring in acid and partially digested fats and proteins into the duodenum, initially that will send an upregulation signal to the stomach, but ultimately it will send an inhibitory signal to the stomach to tell the stomach, hey, we've received the chyme, you can stop making acid, you can stop churning the food. It does this through a nervous system called the enteric nervous system. Please know that name. The enteric nervous system is found between the duodenum and the stomach. And this is the nervous system where the duodenum tells the stomach, hey, you can stop functioning now because we have all the chyme. The second major regulator of stomach function is the chyme itself. When that acidic chyme goes into the duodenum, it stimulates the enteroendocrine cells in the duodenum to make secretin and CCK. So far, you've learned about CCK. That's cholecystokinin, CCK. We learned so far that CCK stimulates the gallbladder and stimulates the pancreas. We will talk about secretin, but just know that secretin's main job is to stimulate the pancreas to secrete HCO3, minus. Do you remember what this chemical is? It's bicarbonate. What is bicarbonate's job in our body? It is to act as a buffer, and we'll talk about why this is so important. For now, you know that CCK and secretin are produced by enteroendocrine cells. So the third point is the pyloric sphincter, and the pyloric sphincter is able to contract and relax to control how much chyme goes from the stomach into the small intestine. You don't want to send off all the chyme into the small intestine at one time because there's a chance that the pH could drop too quickly, too much, and it could create a too acidic environment for that duodenum. Additionally, you want to give the duodenum a little bit of time to work on the present chyme, to break it down, digest the materials, and move it on along through the small intestine. The last point here is on enteroendocrine cells and their job in producing something called GIP. GIP is gastrin inhibiting peptide. And gastrin is the peptide that actually upregulates the stomach. So enteroendocrine cells that produce GIP is going to essentially downregulate the stomach. But in addition to this, there is a second job of GIP, and that is to stimulate insulin secretion. What do we know that insulin does in your brain? What does it signal? Well, insulin signals satiety. So once the food reaches the duodenum, the enteroendocrine cells in that duodenum are going to do two things. They're going to make GIP to inhibit gastrin in the stomach, and they're also going to stimulate insulin secretion to produce insulin to help with sugar regulation, but also to signal to your brain that you are now full. So this slide, I'm going to take my pen and I'm going to star it. This is a very important slide. This is going to put everything into order for you. All the things that you've learned thus far are going to be in illustration form. So let's say you've eaten a steak, high protein. The protein will be partially digested in the stomach, and that steak will also increase the pH of the stomach. The elevated pH of the stomach is going to stimulate G cells, these pink ones right over here. G cells produce gastrin. Gastrin's job is right here. We're going to put a big red box over it. I'm going to put a big arrow right here. Gastrin. In the stomach, gastrin's job is to stimulate chief cells and parietal cells. As a review for you, chief cells make pepsinogen, the inactive form of pepsin, and parietal cells make acid. What do we know that acid does to the pepsinogen? Acid is able to cleave off this portion of pepsinogen to turn it into active pepsin. What is pepsin's job? Well, pepsin's job is to digest proteins. So pepsin goes and partially digests your proteins. And guess what? We're not done here because these guys actually can cycle in to this whole system. Partially digested proteins, amino acids, these guys are going to also act as a stimulus to G cells. So the more pepsinogen you make, the more HCl you make, the more pepsin becomes active, the more you partially break down proteins, and those partially broken down proteins will additionally stimulate G cells to stimulate even more chief cells and parietal cells to keep this cycle going. Your next question might be, well, then when does this whole cycle stop? Because if you keep making more of these two ingredients, you're going to make more enzyme, which means you're going to break down more proteins and you're going to further stimulate more G cells. Well, eventually, this chyme makes its way into the duodenum. It will continue having this cyclic function until the stomach sends its chyme into that duodenum. So this is where we're going to stop on part 2 of digestive system. Please be sure to go over these lectures in order as they do build on each other. And part 3 will be coming up in just a few days.